In this segment, I'm going to talk about polarity genes, segmentation genes, and Hox genes in development. This is a conceptual introduction. The actual genes have different names than the ones I'm going to give them, but the names are different in different phyla and often in different species. And it makes it very confusing to try to understand conceptually how the different genes work. So I'm going to give you an introduction to the ways in which these genes indicate position and control which traits are expressed in which part of a body. For example, why does a leg form in one location, an eye in another, and a heart in another? We're going to start with this hypothetical embryo. It has 12 cells, and it's going to develop first into an anterior with a posterior. It has a dorsal and a belly ventral side. It would also have a lateral left and right side. We won't show that. And in addition, this one is going to have 3 segments. One, 2, and 3. So early on in development, actually before this number of cells, the beginning is anterior-posterior gene expression. And so that would be a gene that we'll call anterior, and another one that we'll call posterior. Now the actual names in different species are all different from this, but they are anterior-posterior polarity genes. I'm showing them in particular locations. In fact, they're sometimes expressed in ways in which the most anterior cells might have high amounts, the most posterior low amounts, and the ones in between would have gradually changing amounts of expression of those particular genes. Second, we have dorsoventral polarity. So here, dorsal and ventral all along here. Second are the segmentation genes, and let me zoom in on one segment here. So here's segment 1, and we have segment anterior and segment posterior. Segment anterior again, segment posterior. So if we look at the whole series, again, each one has a segment anterior and a segment posterior. Segment anterior, segment posterior. The third piece is which segment is which. Those are the Hox genes. Again, remember we have 3 segments, and the Hox genes are expressed from anterior to posterior, always in the same order they occur on the chromosome. So in this case, it's Hox 1, Hox 2, and Hox 3. And same up here, Hox 1, Hox 2. Let's imagine we have a nucleus with a number of chromosomes, and that those chromosomes have a series of different genes on them. If we zoomed in on a particular gene, we'd find that it has a coding region and a regulatory region. So this is where transcription factors would bind to control whether or not the coding region is expressed. So binding here in the regulatory region, and then the coding region here. Some of these genes are transcription factors, and they're involved in development, and other genes are structural genes, or they might be enzymes. So genes for particular proteins that form part of the structure of a cell, or for enzymes that convert one type of molecule or molecules into other types. So let's imagine that we have a series of transcription factors And the transcription factors are the ones we've just been talking about. So we have anterior, posterior, we have dorsal, ventral, we have segment anterior, and segment posterior. So these are the boundaries. And remember that each one has a regulatory region and coding. Now I've shown them side by side. In fact, the regulatory regions can be spread out over multiple locations, and they don't have to be just in front of the coding region. So now let's think about a particular cell. So let's imagine that a particular cell is in a location where, because of earlier events, it has some specific genes expressed. Let's say that it has posterior, it has dorsal, and it has segment anterior. Only for these genes are we getting messenger RNA and the proteins. Okay, so the proteins we have are dorsal, posterior, and segment anterior. So now let's imagine that we've got a series of structural genes or enzymes, and we'll call them A, B, C, D, E, and F. And each of those structural genes will have regulatory regions for particular transcription factors. So let's say this one has regulatory regions for anterior. So this is for anterior, dorsal, and segment posterior. Okay, the proteins that we have include dorsal and segment anterior, which can't bind, and posterior, which can't bind. So we have no gene A expressed. Now let's imagine gene B, and let's imagine that gene B has binding regions for dorsal, for posterior, and for segment anterior. Those are the three proteins that are present in this cell. So we get those three binding. Because those three enhancers bind, we wind up getting messenger RNA and protein B. So we have protein B present in the cell because those three transcription factors are present. And anywhere there are binding sites for those three transcription factors and where there are no repressors to block them, we'll get a particular gene expressed. So let's imagine that here also we have D, dorsal, posterior, and segment anterior. Here also we'll get messenger RNA and protein. Every single cell in that embryo has all of these genes. Every cell has both the transcription factors and the structural protein and enzyme genes. But only some of the transcription factors are being expressed. Only some of them are producing messenger RNA and have the proteins because of earlier events. And therefore, only those proteins, in this case dorsal, posterior, and segment anterior, will be present. And if there are binding sites on particular genes for all three, then we'll get messenger RNA from them. Dorsal, posterior, segment anterior, the proteins bind. That allows RNA polymerase to bind and make the messenger RNA for protein B.